That a bit. Okay, so here is sort of the concept that I'd like to uh, have you think about today, and that is that our various glycoinformatics resources often sort of sit on a little island that's really difficult to get to. Um, uh, we use a variety of tools and informatics approaches which don't actually provide ways for experts in the field to find the glycans that they're interested in and to understand how they're related to other pieces of information. And here's a good example why. We're quite familiar with using the images or the SNFG notation to denote the glycans. But in our knowledge bases and our glycoinformatics resources, we have these obscure accessions and these long strings of glycan sequences, and even this alternative graphical method of describing the glycan structure. But this is all well and good. I mean, certainly this is a fully described glycan structure in terms of the linkages and the monosaccharides. But in addition to being able to describe fully defined glycans, our glycoinformatics resources really need to be able to describe glycans in which we only know some of the pieces of information about our structures. In this case, we're saying that we do know that this is essentially has the topology of a typical complex N-glycan, but we do not know the linkages of any of the ARM monosaccharides or their anomeric configuration. Our sequences and our accessions allow us to describe and to identify those. I just want to hide that. Wasn't what I wanted to hide. I wanted to hide this. Okay. But our sequences can very well describe the fact that certain information is present or missing, but it makes it very difficult to identify the difference between this G27817LK and the previous structure that I showed you, G39764AC. How do we find these different glycans? How do we navigate amongst them? And how do we sort of match them up to the tools that we're using, whether we're talking about analytical tools such as mass spec-based search engines or glycosidases we use in the lab to cleave glycans apart, and all sorts of things. Often the mass spec-based tools give us monosaccharide compositions, typically not in this first form, where we are separating the mannose and gal hexed residues, but more typically in this second form, in which we don't distinguish the different hexoses and hexnacs that have the same mass. But each of these has different glytucan accessions. How are we going to navigate that? And how are we going to deal with the fact that some engines will use a D-hex, others will call it fucose. And then still others will use very stylized abbreviations to talk about the structures that are being identified or are being described. And if we go to our manuscripts, we find language like this long term in quotes, dicylated, biantennary, complex, and glycan. We kind of know what this means, but how do we match this up to the information we have in our glycoinformatics resources? So in our various glycobiology contexts, we have a variety of ways that we use to describe our glycans. We have our pictures. We've got various abbreviations. We've got IUPAC names, and I see a question here about IUPAC condensed notation. I'll point that out in a few spots where we already have access to that. There is prior knowledge. That means that when we describe a glycan in words, we already know which pieces that we're not explicitly describing and what is understood. We often use compositions, and we have sort of named structural motifs, which don't describe the entire glycan, but which describe parts of it or commonly occurring motifs. On the other hand, our portals tend to use accessions, more or less useful. We tend to use sequences such as the IUPAC sequences, but also GlycoCT and works, which give us more precision in how we describe the way that our glycans are made. But often we need to describe when structural details are either missing or unknown. And we need to describe cases where we may not know what the specific, we may not know where a fucose is attached to the structure, for example. So we have undetermined linkage where a specific residue or a substructure might be joined at different places in the glycan structure. We just don't know where it hits. Our informatics portals typically allow us to query by mass or by composition, and we end up with long lists of structures to sort through, often without any great sense of which ones we should care about and which ones we should choose. So many of the tools that I've been working on in this context are trying to address this problem. They're trying to give alternative ways for users to find the specific glycan structures that they are interested in by both making the structural relationships between these structures very clear and by attaching human conceptual semantic names to those structures. And as a group, we'll call these GlyGen tools, but you'll see that I'll introduce various names to these in a moment. Typically, these tools have been relatively loosely integrated with GlyGen. That means that we can have iterative development independent of the GlyGen release cycles. And in some cases, we try to, and generally this is the case, we try to support more glycans than the subset that GlyGen currently supports, which has a very model organism focused strategy. So we try to handle more glycans than merely those that GlyGen supports, but we also generally provide ways to jump back and forth between the GlyGen set and the set that is integrated in these tools. These tools allow us to implement a more complex visualization, user interface, annotation, or alignment, logic, or facilities. And as they mature and as we figure out the best way to use them, they make their way into GlyGen proper. So I'll show you a mix of places where these tools reside, not only in GlyGen and the way that they are integrated in GlyGen, but also how we might jump out to those external tools and then back into GlyGen. Today, I'll try and show you 4 different strategies that you can use to try to find your glycan of interest and sort of explore the space of glycan structures that fit with your research. First, we'll have a look at some structure-based strategies. In particular, the GNOme subsumption-based structure relationships allow us to navigate the structures in ways which try to elucidate which parts are known and which parts are unknown. The Glycan motifs allow us to describe specific parts of each structure and to name them and make them available for search. We have various semantic name-based strategies. In particular, the GNOme synonyms allow us to go from composition strings and other synonyms to a specific structure, as do the Glycan motifs. We have a variety of glycosylation enzyme-based strategies supported by the Sandbox tool, which I'll also demonstrate, allowing us to jump between the enzymes which synthesize the structures and the structures that they synthesize. And lastly, we have a bunch of pure sequence-based strategies where if you have sequences which you don't know the glytucan accession for, there are a variety of ways that you can look them up. Many of these what started out as external tools have been integrated with GlyGen. Subsumption is something that we support quite extensively in GlyGen already. our species annotations incorporate a subsumption logic, which allows us to transfer information about the species of a glycan from its more specific structures to those with less information. We also have information on topologies, compositions, and fully defined structures related to the structure of interest. But we also have these links called related structures and structure browser that jump out to the GNOME infrastructure. And I'll demo those today. Glycomotif gives us a way to name functional parts of structures. It identifies those pieces that are common to many glycans, and it allows us to search for those motifs by name and by looking for the relationship between structures and their motifs and their structures. The Glycomotif infrastructure is also responsible for the type and subtype classifications that you can use on the Glycan search infrastructure. That was demoed a couple of weeks ago. The Sandbox allows us to, or provides glycosylation enzyme relationships with structures, which allows us to look at enzymes, find those enzymes as proteins in GlyGen, and then look at the structures that those enzymes are responsible for helping to synthesize. And then lastly, we have a variety of on-demand sequence searching strategies provided in GlyGen that are integrated versions of tools that you can find externally. So I'm going to transition at this point, after that introduction, to an interactive exercise. This is a bit of an experiment, but we don't have, that many people in the presentation, so I think this can be manageable. I'm going to sort of lead you through a series of steps that I'll hope you'll be able to follow in real time. Pull down the slides from the link in the chat or from the link you can see here. And I recommend downloading it rather than using it within the box framework. Each of these slides has embedded links that will help you catch up if you get lost. But if you click on those links directly, you're going to lose the benefit of the exploration aspect of this, this demo. Yes, I can put the link in the chat again. Here we go. So ideally, you can, I will try and sort of tell you in words what to do. I'll give you a couple of minutes to do it independently, and then I will demonstrate it as part of the presentation so that you can see how I would tackle it, which won't always be the same method that you will use. But hopefully we'll get to somewhat the same place, and there are lots of opportunities there to recover. Okay. I'll just give a minute or two for people to find those oh, we have Q&A. What is subsumption? Oh, yes, I see a question about subsumption. Let me answer that right up front because that's a really important one. If we look at the structure here on slide 3, in which all of the anomeric configurations and the carbon bond linkages are described, you'll see we've got the alphas and betas. We've got the 2, 4, 6, the 3 down here for the mannoses, etc., etc. These describe how the glycan is connected to, how the monosaccharides in the glycan are connected and linked to each other. This second glycan structure I'm showing here has a whole bunch of question marks in various places for the anomeric configuration on these 2 antennae and for the carbon bond linkages. In some sense, this is an accurate description. Oh, and it also leaves out the anomeric configuration at the reducing end. In some sense, this is this description of the potential glycan structures represented is more broad. It allows more structures to be described, but it's also a way of being more precise about what we know and what we don't know. So in this case, we don't know the linkages on the arms or on the antenna, and so this structure subsumes the more specific one. That's the use of the terminology. I also, Renee is prompting me to talk about composition. So in this case, we have a composition representation, which is even less information about the specific structure. These compositions here also subsume both of these structures, both the one with lack of information on the antenna and the one with fully described linkages. I hope that answers the question on subsumption, at least for the moment. We're about to transition into a bunch of tools where we'll see some of this being demonstrated more explicitly. So hopefully that will help reinforce things too. All right, I think that's given people enough time to find the slides. So let's jump into the demo. I'm going to go to GlyGen and find the page corresponding to this Glycomics session. And for that, the simplest way to do it is just to type it into the global search field. You'll see that it says that there is a Glycan exactly matching that accession found. And if you click on that, that link, you will end up at this page. In my slides, if you click on the hyperlink in the heading here, you will also jump directly to this page. Okay, so this is our standard GlyGen landing page for Glycan structure. You can see that this is the structure that I pointed out earlier that is missing some information on the antenna here and at the reducing end. I want to show you first a little bit where some of these concepts of the relationships between the glycans, the motifs, and the various other pieces of information are integrated in the GlyGen page. If we click on the organism section, we jump down to this area of the page, which shows us a variety of species: human, mouse, pig, rat, etc., etc. And you can see that there are badges under each of these describing where the evidence for this species annotation came from. In particular, you can see that many of them, most of them in fact, have an annotation called subsumption. This means that there is a structure with more specific information which has received this human annotation directly. And we are propagating that annotation from the more specific version of the structure up to this less specific version of the structure. If you look at a variety of these, I wonder if anybody can spot a Glycomics session that seems pretty common between many of these different species annotations. Let's see, 7ZM. Yeah, 7ZM is there in a lot of spots. We can actually click on there and navigate to it. In fact, why don't I do it? Spot. Here is that structure. Which we will see soon enough. Here we go. The image will come up shortly. So this must be an oh no, there it is. Just some delay. So you can see that in this particular case, this is the alpha-6 linked sialic acids, the 2-arm over here, and without specifying the reducing end anima. So this is not fully specified, but it is mostly specified. And we can see if we look at the organism entries here, we have specific entries for this structure from Glycosmos for each of these different species. So subsumption is used to propagate the annotations from more specific versions of the structure to less specific versions of the structure. We will actually see, where is it? The 84467IZ later. It is a fully specified version of the structure we just looked at. Okay, so then the next place I'd like to give you a tour of is the motifs. This is a section that describes the motifs which align to this structure. These provide both information about important aspects. You can see that even though we don't know the linkages on the arms, two different motifs which represent different styles of linkages match. So the motif alignments here are subsumption aware. You can also see that we align to, we have the 3-sialyl motif alignment here. Again, we don't know this for our structure, but it is one of the possible instantiations of the linkages. And so we get to keep that too. Further down, we have a list of the biosynthetic enzymes. These describe the enzymes which are annotated as are responsible for the biosynthesis of this structure. You can see, the accessions. You can jump into the GlyGen page for these proteins, and you can also jump over to the Sandbox to investigate this structure. We will do that later, but we won't do it just yet. Lastly, I want to show you the subsumption section of this page. The subsumption section shows us information about structures with less information at particular levels of understanding or levels of characterization, from the so-called base composition without any stereochemistry, the composition level in which we have stereochemistry of our hexes and our hexnacs, and the topology version in which we have no anomeric configuration or carbon bond linkage information, but we do have the information about how the monosaccharides are linked together. In addition to that information up the subsumption relationships, we have information about the 19 different fully defined structures which are underneath, more specific than our current structure. You can see that each of these fills in all of the linkage information, but now we have a different problem. How are we going to choose which of these we like after looking through all 19 of these? It's actually quite difficult, and if you're old like me and your eyes are getting bad, it's really hard to sort of see exactly what the difference is between these different structures and whether you should consider them to be useful and a good match to what you're looking for. So as a way of solving this problem or helping us understand the relationships between these different structures, and at this point, I'm about to transition to this slide. This is slide 18. Is everybody hanging with me okay so far? All right, I got at least one thumbs up. I'll take it. Okay, good. Okay, so we're going to go back up to the top of this Glycan page for G27817LK. And we're going to click on this button called Related Glycans and choose GlyGen and Glycans. And again, if you look at the next slide, slide 19, this link in the header will take you directly to this page. Okay, so we have jumped here into the GNOme interactive browser that represents the subsumption, the more or less information relationship between glycan structures in a graphical and a navigable way. The browser will zoom as needed if you've got bad eyes like me. This is our Glycan of focus. And I'd like to challenge you to start at this Glycan and to look at the more specific versions of the structures to the right and gradually navigate down the tree until you can find the fully defined alpha-6 linked disilated biantennary complex and glycan structure. It's there. It will take a few steps. I'll give you a couple of minutes to work on it independently, and then I will demonstrate how I would do so. The structure I'm expecting you to find is the G844-6Z67IZ, which you can find linked here. But again, if you directly just click on the link, you'll lose the benefit of the activity of learning how to navigate through the subsumption hierarchy using GNOME. The basic strategy here is that you should think about the structure you're interested in, look at the structures to the right to see whether or not they have more information of the sort that you would like for your end glycan, in this case. In this case, we can see that the one at the top instantiates the beta at the reducing end. The middle one instantiates the alphas for the sialic acid, and the one at the bottom puts a 3 for the sialic acid linkage. So we don't want the one with the 3 because we're interested in 6 linkage sialic acids, but either of the other 2 would be fine as a starting point for navigating to something more specific. Let's look at our next step. We could choose to add the betas. So I chose to add the alpha at the for the sialic acids. It looks like we could choose to add the betas for the gals. Zooming in again. Here we have alpha 6. That looks promising. That's definitely something we want. Continue to navigate down the tree. 2 beta 2 linkages for the GalNAcs looks promising. Here I'm instantiating the beta at the core. And lastly, I can go to the structure, which is fully instantiated. We've got the beta 2 linkage on the 2 GlyCnacs. We've got the alpha 6 linkage on the sialic acids, and we are good to go. Having discovered the glycan that we're interested in, we can click the right button and jump back into GlyGen by selecting GlyGen from the links part of the menu. Okay. Renee is asking me why there are so many structures with missing information. The why is because people are allowed to describe glycans with whatever information they have. And in many cases, we have lost information because our characterization strategies do not tell us exactly which linkage we have for these arms, or what the linkage is for the sialic acids, for example. Also because people are able to describe their glycans in words, and sometimes that doesn't communicate specific pieces of information, and so on. Yeah, so thank you for the comment, Tyler. This particular right click is expensive because this particular set of structures is very, very large. There's a lot of structures with this composition. And I don't know if you can see, but one of the options here is a way to select a list of all of the Glycomics accessions which are less specific than this particular structure. It takes a long time for it to do that work to collect all of that accessions. This particular composition and set of structures related to it is one of the largest that you can hit. So unfortunately, it does take a little bit of time for that to happen. Okay. Other questions or comments? Okay, so here we are at the fully specified version of this glycan. I want to point out, as I did before, if we navigate down to the organism section, we can see that there are much more specific annotations to specific glycan databases that provide these species annotations. This is because this is a fully described glycan, and there is a lot more direct annotations of fully described glycans, even though we have all of these glycans with missing information that we need to navigate and understand. Okay. The next thing I'd like to explore, now that we're on a fully defined glycan, I'd like to explore at the top of this page the Sandbox button. Let's click on the Sandbox button, and we will end up in the GlyGen Sandbox for this accession. Let me find which slide I'm on. So I'm on slide 22, and again, the hyperlink at the top here. Whoops, sorry. No, slide 21. The hyperlink at the top will take you to this page directly. So this is the Sandbox. The Sandbox allows us to click on different residues to see what enzymes are responsible for, or potentially responsible for, adding those monosaccharides to the structure. You can click all over the glycan and see how that see what enzymes might be responsible. You can also highlight residues based on their relationships. So you can see that the relationship structure of the monosaccharides is fully described. The other really nice thing that the Sandbox allows us to do is it allows us to look at structures which are related to the current one by the addition of new monosaccharides. I want you to scroll down until you find the monosaccharide with the addition of an alpha-3 fucose to one of the GalNAcs, the upper arm GalNAc, the alpha-6 mannose arm GalNAc. Its glytucan accession is G63565GW. And if we double click on that, the Sandbox will shift to focusing on that new structure. As before, we can click on all of the residues to find out what enzymes are responsible for adding those monosaccharides to the structure, including the new residue that we just looked at. In this case, we have, in particular, both the mouse and the human enzymes mentioned. Okay. So as you can see, the alpha-3 fucose is attached by, at least in humans, by food 3, food 4, food 5, food is that a 8 and 9, or is that a 6? I need to wear my glasses. Apparently it's a 6. And we can then navigate to GlyGen or to UniPro in order to understand more about that gene and how it is involved with the pathways for constructing glycans like this. We can jump either directly back into GlyGen with this GlyGen data link, or we can click over into the GNOme Subsumption browser in order to figure out perhaps Here's a question. Maybe we like this structure, but we don't actually know which arm this fucose is located on. Let's navigate back to the GNOME Subsumption browser and figure out a structure which precisely describes the case where we don't know whether this fucose is attached to the top arm or to the bottom arm. I'm going to click on this GNOME Subsumption Navigator section. Okay, so we have transitioned again. We went through our second Sandbox place and looked at the fucose that was there. We clicked on the GNOME link, and we ended up at slide 23. Here we have the structure we started with, fully defined with the fucose attached to the upper arm GlyCan. So let's figure out if any of the less specific versions of this Glycan describes the case where we do not know which arm the fucose is attached to. The top option here is a partially correct answer. It's partially correct because the mannose linkages here are not specific whether they're 3 or 6, but notice how much information we've lost about the fucose and the arm linkages. Perhaps we can do better. Let's look at the second option. The second option is fully defined with the alpha-3 linkage, but this bracket indicates that we do not know which monosaccharide this fucose is joined to. This is still a loss of too much information because this allows this fucose to be a core fucose, for example, which may be something we specifically know that we don't have. Let's look at the third option here. The third option, again, loss of too much information. In this case, it is specific to the upper arm. Lastly, let's look at this last case. This also loses a little bit more information than we might like because it does not specify the linkage of the sialic acids. Nevertheless, it does make it clear that we don't know which arm with the fucose is joined to which of the mannoses. So of the options to try and decide how to describe our glycan such that we don't know which arm this fucose is attached to, I think this one down here is probably the best choice. Okay, just checking for questions or comments or anything else. Okay. I'm going to jump back to our GlyGen page for G84467IZ. And I'm going to show you the feature view. The feature view is integrated in GlyGen. It allows us to investigate the placement of the motifs that are annotated for this structure, such as the type 2 LACNAC, or for the enzymes, the specific residues that those enzymes are related to. For example, we can ask the question, which residue does ST6-Gal1 help synthesize or add to the structure? What residue does ALG1 attach, or ALG14 and 15, 14 and 13? We can investigate these structures easily and the relationship between the enzymes. This information is being drawn from the Sandbox and is presented here in GlyGen for us to explore. So if we wanted to discover what the MGAT1 enzyme does, we just need to click on this box to see that it attaches the alpha-3 arm beta-2 GlyCan residue to this structure, whereas MGAT2 is responsible for the alpha-6 arm beta 2 GlyCan. I want to take us to a slightly different structure, and I'll do that by going back to the GNOME page and looking at it's not quite the one I want, but actually I'll do that from here, otherwise it won't match the this one here. So this is slide 25, G01510MQ. I chose this just because it's got a bit more interesting structure to it. Okay. So this is a similar type of structure to the one we were looking at earlier. It does have a gal-nac rather than a gal at this position of the structure. I'd like to show you in the feature view that our motifs now are richer because we have more interesting things going on. And in particular, notice that our, okay, we're still being slow. I guess we're still being slow. Okay, well, we can see in the slides that our motif highlighting and our motif alignments are able to go over the undetermined linkage to identify that as a putative motif alignment site, even though the explicit linkage is not available for this structure. Okay, I'm going to open this link directly. So I went and let me show you. Let me see if this is now caught up. No, I guess JavaScript is slow. You can see on this pop-up that we can jump to the details for this motif by going to the GlyGen page, like so. And what it will show us in a minute is the structure itself. In fact, oh no, I don't have a shot of that. All right, so while that's coming, I will move on and come back to that. I want to point out that you can access the GNOME structure browser from the home page of GlyGen. You can see it here as one of these tiles. And in the slides, I'm suggesting that you can try to do the same activity we did earlier to find our diacylated biantennary complex N glycan, which is equivalent to this composition string, and navigate down to the structures of interest. So let's do that. Again, in the interest of time, I will just do this directly. You can see we land here at an empty page. In order to build an N-glycan structure, we use these arrows to add to our composition. We need at least 2 GlyCnacs. Then we have 3 mannoses, followed by a couple of more GlyCnacs, 2 gals, and then 2 sialic acids. Notice we now have a variety of different topologies, some of which are N-glycan related and some of which are not, which are consistent or not consistent with the structure that we're interested in. If we click on the structure that we that is most closely related to what we're interested in, we can then see the structures which are related and have less information. This one here looks interesting, and we can navigate on down until we get to a fully defined structure of interest. In this case, I've chosen one with an alpha 3 and an alpha 6 aunt. Notice that we can jump back to the composition area, and if I reset things, we can even search for a glycanic session or search for a composition string, such as one you might get from Bionic or a similar tool, which will jump us directly into this spot. We can also define URLs, which define the compositions that we want to see, and jump directly into this spot in the same way. All right, so let's go back to our motif example. No, let's try again. All right, so here is the image for the motif, some information about it, some keywords. and some, variety of information there. You can see down below, we have all of the structures with their highlighting that indicate where that motif aligns. We can also see that there are 266 different structures which this motif aligns to. That's actually a really nice result. We could look through them by hand if we like. We can also type that into the global search in order to do this more directly. If all of these motif alignments are available as search terms for our global search. And what you'll notice is we'll end up with 266 different structures matched in the Glycan set. The nice thing about this version of the search result is that we have all of the sorting and filtering strategies available to us. So, for example, we could look down this list and observe we're getting mostly N-glycans, but eventually we might see some O-glycans that we're uninterested in, or perhaps we're interested in those that are not N-glycans, and we can select those directly. All right. In the interest of time, I'm going to skip to the final little sort of vignette I'd like to share with you. This is slide 28. I'd like to show you that GlyGen is able to handle sequence-based searching. So the way that we get to that is we go to the home page and we ask to explore glycans. You can see two different tabs here, Substructure Search and Structure Search. I'll demo both of those quickly. If we paste a GlycoCT sequence into the sequence area, in this case, I'm going to restrict the results to the reducing end, and I click Submit. In this case, I'm essentially reproducing the search for the 3-silyl lacnac motif that was that we were just looking at. I think. Oh, okay. So it doesn't like the fact that the new lines were lost when I pasted it into PowerPoint. So I'm going to go to this link here, G01. I've put it in there to jump directly to the digital sequence, copied it to the clipboard, and I'm pasting it now as proper GlycoCT. And you can see we have a list of the glycans which have that, that substructure aligned. In this case, I got the substructure as from the motif page, but we could use any structure we like in order to do that search, as long as we can describe it with one of our sequences. Similarly, we could do a search with works. Here, I'm going to do a structure search. In the notes, I have the works sequence, and I will paste it in. Here I'm doing a structure search. This is subsumption aware. This will find structures which match this sequence or which have less information. So you can see that there are 22 different structures which match this, although the one that we're interested in came up first. But you can see that these structures all match with more or less information. The reason why we got both the alpha and beta is that the sequence that we supplied was actually missing the anomeric configuration at the reducing end. So this is a subsumption-aware version of the sequence search. But lastly, I'd like to show you that we have a way of doing an exact sequence search. This is using a tool from the Glycomics tool set, and it allows us to paste in
Good. How are you?
a glyc2can sequence
I take a burrito.
or a
Wheat.
work sequence, submit it to the tool, and it will look it up to see whether or not it has been previously registered in glyc2can and whether it has a glyc2can accession. All right, so here are some final thoughts. These tools give you lots of ways to explore and find your glycans of interest. The subsumption concept, the enzymes, the biosynthetic enzymes, the motifs are all baked into the GlyGen portal directly, but there are also these external tools which are linked in, which offer additional navigation and exploration opportunities. We have lots of creative solutions to your glycoinformatic challenges, and we'd love to hear about your challenges so that we can help you solve your glycoinformatics problems. We may not be able to do them directly within the GlyGen framework. Sometimes it will take external tools, but often we have already confronted problems such as the ones you're hitting, and we've got strategies for trying to solve them.